Okay, I don't know why they like the second name. Huh? Totally?
I don't know. Okay. So Lejeuneae is one of the largest genera of liverworts. So in the index Hepaticum records approximately 1,700 names worldwide under Lejeunea. And Lejeunea are very common in the tropical rainforest, where they have successfully adapted to the various epiphytic habitat in the forest, like tree trunks, tree bases, and in spite of the abundance in the tropical forest, the taxonomy of Lejeuneaceae species is still poorly known and in the state of chaos. Why do I say in the state of chaos? Let's go back to the history of Lejeuneae itself. Okay, as you can see in this figure shows the numbers of species described in Lejeuneae from 1820 to 2000. So the figure clearly demonstrating that most taxa were described in the past century. So the first major increase in species number took place between 1814 to 1849. And this can be explained by the publication of Synopsis Hepaticum by Kocher. Kocher described almost about 300 species of Lejeunea, and most of them are from tropical. And in this stage, in 1840 to 1849, most of the hepatologists consider this as the golden period of hepatology. Okay, you can see from the figures also, the second important rise in the number of species was in the decade of 1880 to 1889. And this is due to the contribution of Richard Spruce. Richard Spruce's work. Richard Spruce is a very amazing British hepatologist. He was, in the opinion of some, the most outstanding explorer in South America. What happened is, he, at the age of 32, he decided to travel to South America, where he remained in the Amazon jungle in Andes for 15 years. So out of these years came his wonderful work, that is Hepaticum of Amazon and of the Andes of Peru and Ecuador. Here they came out with a very wonderful description of Lejeunea itself. So another author that I want to share who contributed to the rise in the number of Lejeunea was this person, Frank Stefani. So Frank Stefani described about 2,000 species under the Lejeuneaceae family in his 6th volume of Species Hepaticum. What happened is, Stefani created a lot of synonyms, described the same species under different names, and this has caused a lot of confusion. So, and Grestein considered this as the most dramatic event in the history of Lejeuneaceae family research. So therefore, with the chaotic situation happens in Lejeunea and in the family of Lejeuneaceae, a comprehensive study of the genus Lejeunea has been long considered a priority for virology research in this part of the world. So as for me, frankly speaking, I'm not able to finish a world monograph of Lejeunea. Even if I'm given 2 or 3 lives again, I think I will not able to study this genus. Perhaps, I think the problems that are created by Frank Stefani is never ever can be solved. So the work is just too huge with a lot of chaotic species created by Frank Stefani. So therefore I choose Malaysia because I'm a Malaysian. So this map shows the number of species recorded in each country. Okay, from this we can see China recorded 37 species of Lejeunea, followed by Japan, Indonesia 23 and 10 respectively. Okay, let's see in Malaysia. Ephraim 1937 was the first to record the species of Lejeunea in Sarawak, followed by Mizutani in 1960 and 1970 recorded 23 species of Lejeunea in Mount Kinabalu. And Kodama, 1976 also recorded one new species from Mount Kinabalu, followed by in Peninsular Malaysia, Inoue, 1967, described the oil bodies from Cameron Islands, 4 species. Kitagawa, 1969, 1 species from Penang Hill. And Tixia, 1971, 1980, 3 species. So total of Lejeunea reported from Malaysia is 29 species. What happened with all these 29 species? That they only provide short notes and very primary keys. So good description and illustration are lacking. And ecological and distribution data on each species are very scanty. They did not describe all this. So the need for detailed taxonomy study of Lejeunea in Malaysia is very urgent and desirable. So I took up this genus in Malaysia. So my objective on my revision are to first identify the species of Lejeunea in Malaysia, prepare a detailed identification key to the taxa of the genus in Malaysia, describe, illustrate in detail all the taxa of this genus in Malaysia, and address the taxonomy problem, and also lastly examine the phylogenetic relationship among the taxa using morphological data. Okay, what happens after 30 years Lejeunea in Malaysia? This is derived from my PhD results. Okay, in the beginning only 5 states that recorded as Lejeunea. After my study, I found out that most of the states have also Lejeunea. Okay, so total reported from Malaysia now is 32 taxa. That means 30 species and 2 varieties. Okay, from my projects, the genus in Malaysia comprises 32 taxa, among which 3 new synonyms, 2 new combinations, and 3 new species were described. And also 11 species are reported first time from Peninsular Malaysia and 1 species from East Malaysia. Okay, from my study also, I found out new data. They also underused morphological characters used for the first time. Superior center cells, it shows 4 cells here, 1, 2, 3, 4, and 2 cells. Which this is the new and very stable taxonomy features of Lejeunea, which we never used before for this time. And I found it that in this character is very stable, and it can be used for classifications and can be used for subgeneric classification of circumscription. Next, critical of the leaf lamina cell. I'm using STM to scan through all the 32 taxa, and I found out there are 2 distinct. One is the papillus, and one is the smooth cuticles. However, in 2012, there's one paper on Lejeunea revealed the presence of surface wax. They believe that this is the surface of wax by soaking the specimen in chloroform. So I have not tested this. I'm thinking I will further my study on this. Okay, oil bodies. Oil bodies can only be seen in fresh specimens. So it will disintegrate. So we have to look on the oil bodies very carefully in the field, or after we collected from the field. Only within 24 hours we can see the oil bodies. As you can see here, I have found out identified 4 types of oil bodies. One is granule type, one is papillus type, one is in between of these 2, and one is shining and glistering type with smooth surface. Okay, I want to explain more on the disintegration of the oil bodies, because in my project, most of my specimens are from fresh samples, where this is one of the advantage of study of fresh samples, where you can observe the oil bodies. So I observed these oil bodies. I found out there are 2 types of disintegration happen. The first type is merging of the granule together and eventually forming a bigger granule. The second type is by merging of the granule together and followed by a burst of all granule. That means the small granules are seen free within the cytoplasm. I believe all of you are very puzzled, do not understand what I'm talking about here, right? So I'm show you some of picture. Okay, this we consider this as one oil body. Inside the oil body, there are smaller granules. So I've explained the first type is the the whole oil body will combine together, and the second type is they did not combine together, but they will burst up, and at the end it will become like this. Okay, I'll show you this video. You can see the process of the integration. You can see carefully here this type and this type. You can see they started to combine. The smaller granules started to combine to become bigger granules. They become bigger and bigger. This is the oil body. You can see the smaller granule inside the oil body. Now you can see it becomes one. So this is one type, the first type of the disintegration. Okay, now you can see this is the second type, where the smaller granule inside the oil body, they did not combine together, but they will have one burst where all the small oil bodies will come out from the original oil body. Can you see that? This is the second type. It's very different from this. This is the first type, this is the second type. Okay, that's all. Okay, the last new data is vegetative propagules. I found there are three types. The first type is ribbon-like propagule, and the second type is the plantlet regenerant, and the third type is the specialized branch groups of these plants. So I want to share about this species, the vegetative propagule by specialized groups of branch. This vegetative propagule is very special. It only grows in Lejeunea dimorpha. Lejeunea dimorpha is a new species. It's a new species described by Kodama from Mount Kinabalu. So Kodama described, as you can see here, this vegetative propagule is very different from the original plant. As you can see, this leaf is in reniform shape and is bigger than, we call it underleaf, the third leaf, the underleaf. This is the original plant, and it is smaller, and the form is very different. And you also can see the margin of the branch. The margin of the branch leaf have this, they call it rhizoid, hyaline rhizoid. So what happened is, when Kodama described this species, and this species is based on the dimorphic leaf and underleaf of the plant. You see, Kodama saw this difference, so he named it dimorphic. But in his description, he mentioned the most remarkable feature is the curious dimorphism presented by leaf and underleaf. Kodama did not understand why this special branch grow in this plant, and he just assumed this as a new species. So, however, in Poch et al. 1995 discovered that this special development of dimorphic leaf and underleaf are due to the vegetative propagule, where the plant produce specialized branches with crenulate leaf This is the crenulate leaf, and the marginal rhizome, and a very large underleaf. So they also further explain that the specialized branch are actually a mass vegetative propagule, which disperse by breaking off and leaf lobe with the marginal rhizome, and the marginal rhizome will sprout and develop new plantlet. When I read this article by Poch, I cannot understand, I cannot imagine what is happening over here. Even though I have the type specimen from Lejeunea Lib from Kinabalu, but I just can see this plant attached to this plant, and I cannot understand what Poch is talking about. So I brought this article and explained, and I discussed with Vincent Daman. So we try to picture what is happening around, but we cannot finalize what Poch is trying to say. I also bring this case to discuss with my friends, and they say, hey, this is just a speculation, and it never happened, it never seen or happened in other Lejeunea, or maybe Poch just speculate these matters. But I really thank God that I saw this very amazing growth way when Encik Daman brought me back a specimen from Chama. So a lot of them, as you can see the picture that I attached here, this is the vegetative propagule. You see the rhizome attached to the host, the leaf. Oh, sorry. The rhizome attached to the leaf. The purpose. This is for attachment in the host, and this vegetative is started to rot, and the new plants started to grow from here. Because in the previous specimen that I received, and it's also the type specimen that I received, I did not see this incident. So only from specimen from Chama, I see a lot of them like this. So this is a very amazing incident happened in this species. So from my result, 32 species are illustrated. Morphologic character I discussed in detail. All the specimen examined are listed in full. I also prepared the distribution map and also identification key to the species. This example of my illustration is some of the habits of Lejeunea. And lastly is the parent characters. Encik Daman once told me that liverworts is the word liverworts is if you believe that it can cure your liver. In those times, like in World War, they used to consume or they used to eat liverworts in order to cure their liver or their heart disease. But for now, I think if you have liver problem and heart problem, please don't consume liverworts. Please go and see doctor. Okay, so this is only one part of my project. There are few parts that I did not share to you, like the phytogeography and also relationship among them. So with this, I thank you. I would like to take this opportunity to thank Dato' and also Encik Daman for helping me a lot, encouraging me a lot to study liverworts. I think without them, this cannot be a success. Thank you very much.
Thank you, Dr. Lee. The credit should go to Daman to understand liverworts and mosses. My understanding of liverworts and mosses were due to, I think, our former teacher, Dr. Kuahara. Well, I studied under him in the 70s, and Daman must be in the 80s, Kuahara. And as you notice, the earlier work were mostly by Japanese, other than one or two texts here and other. Because the Japanese, apparently, there are so many hepatologists and also muscologists from Japan. And Kuahara came purposely to teach us about bryophytes in UN universities. So, to the student, this study really illustrates to you how Dr. Lee conceptualized her PhD work. Realizing that there are so many species of Lejeunea, and the taxonomy was a little chaotic. Of course, you notice that many problem taxonomy are almost like that, whether small or big, there are problems in taxonomy. I think, for example, Hussein, when he studied Vatika, realized that calyx morphology seemed to be very, very significant for taxonomy. And Dr. Lee more or less saw three things. You know, the oil bodies. My God, some people can put video in presentation. I just don't understand how to do it. The oil bodies, and also the so-called cuticle cells, and also the superior central cells. So these are at least three taxonomic characters which were ignored by previous people, and she highlighted this thing in her taxonomic work. This illustrates what does a master's or constitute in enhancing knowledge. You know, if you take everything for granted from the literature, from everything, then you don't enhance or you don't increase knowledge, ilmu pengetahuan. So when you look further into one case like Vatika or Lejeuneaceae, you may add new characters, new understanding, especially the case of Lejeuneaceae dimorpha, which I might call trimorpha now. You know, but if not because of the specimen which Cik Daman brought from Gunung Chama, she may not be able to unravel, unresolve the so-called hypothesis by thought. So this is what I call, you know, students of taxonomy, students of ecology, students of science always look forward into discovering, understanding new things. So she has added 3 new species and reduced couple into synonymy, and by elucidating, by bringing out new characters, and we thought she had added new knowledge. And her excellent examiner was a professor, a very outstanding professor from Germany, now residing in Paris. And he, I don't know, I've never met him, must be very senior now, granted her thesis has been one of the excellent theses in the field. You know, we congratulated Dr. Lee. So now she wants to continue further to understand liver works as a postdoc here. So the floor is yours before we eat something behind there.